Innovation is held back mostly because of our lack of imagination. If you look at the innovation in drugs, innovation has closely followed innovations in analytical testing. The more we are able to measure properties of some material, including its effect on the body, then the better we are at devising a proper material that does what we need it to do.
Welcome back to the Smart Biotech Scientist. I'm your host, David Brühlmann, and today we are revolutionizing downstream processing with Scott Wheelwright, who is the chairman, CTO, and co-founder at BioChromatographix International. With decades building companies like Biolnno Bioscience and Innovent Biologics, plus leadership roles at Abbott and Chiron, Scott is now rethinking chromatography from diffusion to convection with BCI's Access Flow platform. Let's explore how next-generation chromatography is transforming bioprocessing. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Scott, to the Smart Biotech Scientist. It's good to have you on today.
Thank you. It's nice to be here.
Scott, share something that you believe about bioprocess development that most people disagree with.
A lot of people are conservative about what's going to happen and what's possible. I believe that there are many opportunities, particularly to reduce the cost of manufacturing of biopharmaceuticals. Some of these new therapies are extremely expensive, and I think there's a lot of opportunity for people to spend some diligence and really delve into what are the physical limits that lead to these current costs.
I very much looked forward to our conversation and to talk about these innovations and how to progress further in bioprocessing. But before we do that, Scott, draw us into your story because you have decades of experience, and tell us how you got started in biotech and the journey along the way, founding various companies and now recently co-founding another company. So I'm really excited about that.
Well, thank you, David. I'm very grateful to have been part of this industry. The healthcare industry is really a great industry because the people that are in the industry are here because they're nice people. They want to help other people. And you can think of some other industries where they focus on money and power. But in this industry, people focus on helping others. And it just makes it really pleasant to be part of. I'm very grateful. You know, when I graduated from graduate school, I was not able to persuade. You know, the industry was just starting out. This was 40 years ago. And I was not able to persuade the startup biotechs that they needed an engineer. They thought that, our biochemists are really good at purification. And I said, well, they don't understand mass transfer and heat transfer and these things. And they said, we don't need to worry about that. And I said, well, you will someday. I went to work for Abbott because Abbott had a large engineering division. I spent a lot of time as a translator between the engineers and the scientists, helping the engineers understand what they needed to know in order for us to be successful at bringing products to market. I was fortunate after leaving Abbott to work for several biotech companies in the Bay Area. Chiron, which was later acquired by Novartis, but Chiron was one of the first biotech startups in the San Francisco area, and Sios, which was acquired by Johnson & Johnson. But we had some great projects. At Abbott, we developed the world's first commercial diagnostic for HIV. We beat the second place company by 2 weeks. The second place was a company called Electronucleonics. You've heard of them?
I've not heard of them.
No, nobody's heard of them. They were second. and we made hundreds of millions of dollars protecting people's blood. Arthur Ashe, the great tennis player, he would have lived if we had developed our test a few years earlier. It's very unfortunate, but at least we were able to save many millions of people's lives. And then at Chiron, we did the world's first diagnostic for hepatitis C. This is an interesting. Hepatitis C is the only virus discovery that's ever been attributed to a private company and not to an academic or research organization. Our project manager, Michael Houghton, was awarded the Nobel Prize a few years ago for that project. At the time, I never thought it would work. I never thought he'd get it, and we did. Who knows? You're just very fortunate. Sometimes it's luck to get into the right position. A few years after that, I was working with some friends in California, and the Chinese government opened what they called a returnees package, 1,000 talents. If you were Chinese born and were willing to go back to China to work, didn't matter what your passport was at the time, they gave you free money, about $100,000 to $200,000, and you could do whatever you wanted. And so many of my friends went back, and they invited me to go with them, although I wasn't Chinese born. And they needed help on the manufacturing side. Mostly these people were researchers. We had a great time. I spent 15 years in China.
Wow, that's amazing. Wow, what an experience.
Yeah, I feel very fortunate. It's a great industry, a great career, just opportunities everywhere.
And now recently you have co-founded another company in Singapore.
Right. So my original PhD work was at University of California, Berkeley, under Ted Vermeulen and Gerhard Klein, who wrote the section in the Chemical Engineer's Handbook, Chromatography and Adsorption. And I'd always had a soft spot in my heart for those technologies, but my career had been focused on manufacturing and reducing costs and getting products to market. But I decided it was time to, I was talking to some friends who were chromatography experts in Europe and in Japan. We had an interesting conversation at a technical conference actually in Vietnam a few years ago. I think the time is right for us to explore this new technology and come up with a new opportunity for the industry that would really help these new advanced therapeutic medicinal products, these ATMPs like cell therapy, gene therapy, mRNA, all of these things. And so we did. So we just decided, well, let's do this in Singapore because frankly, my wife wanted to live in Singapore. And, you kind of do what your wife wants you to do. And so we moved to Singapore and we started this company. I'm very fortunate to have a great co-founder, Shirley Ho. She is a marvelous marketing expert. I've known her for a dozen years. We've worked together on various industry projects, never in the same company. But I thought first thing you need is marketing. You need somebody who understands how to sell the product. You can't just spend years developing it. You'll spend a lot of money and it may not be what the client needs. So that's where we are.
Yeah, that's absolutely key to start with the end in mind and especially with the customer in mind when you're developing product. And I've seen too many scientists actually not understanding that, building a product that nobody wanted, but it was scientifically and technologically interesting, right?
Yeah, that's a challenge. I've always felt that way about process development. So in process development, it's important to understand what the goal is. Process development is very interesting because the challenges are very different from those of research and the battle that we engineers, process people, we fight all the time against the researchers. The researchers are very interested in the technical aspects and understanding every detail. And as an engineer, my goal is to get the product to market. We want to make money. I mean, that's how we're going to succeed. We're not going to succeed if we go broke.
No. Speaking of challenges now, as we're looking into the technology, what current challenges do you see in purification and what are some challenges holding back innovation?
Innovation is held back mostly because of our lack of imagination. If you look at the innovation in drugs, innovation has closely followed innovations in analytical testing. The more we are able to measure properties of some material, including its effect on the body, then the better we are at devising a proper material that does what we need it to do. And I think that's true of the bioprocess field and production of biopharmaceuticals. There's a big challenge, and as new materials come out, actually there was a new invention brought to my attention just this week by a company that does 3D printing. So we use 3D printing in making our devices for chromatography, for purification, and certainly for prototyping. There's nothing quite as fast and so simple. But now this new 3D printing for making molds for extrusion, not for extrusion, but for forming of plastic parts and so on out of cinder ceramic. And they're highly precise and so much better. And it used to take $10,000, $100,000 for a mold, and it had to be made of steel, and it would take months. And now you can do it in days. It may not last as long as the steel one, but you can do the prototypes. You can get it just exactly the way you want it and make sure it works. These new materials are just amazing. Part of what we've done with BioChromatographix is new materials, new discoveries in how to use these materials and how to control them in order to make a new solid phase that can be used for separating viruses to a higher quality than we could ever do before. It's really kind of wonderful.
How does your technology work? Because what I understood is that you're rethinking chromatography from diffusion to convection. Can you elaborate a bit more on how that works?
So classical chromatography has been around for over 100 years, maybe close to 150. Developed by some people in Sweden, and it's developed many years by the groups in Uppsala. And it's based on relative different adsorption or sticking of a molecule or a nanoparticle or whatever to a solid phase. So it's a liquid phase. You have your sample in a liquid phase. You have come in contact with a solid phase. Your material preferentially goes to the liquid phase, and then we can separate how it comes off. So we can control the conditions of adsorption and desorption. And so we can separate materials even if they're very close together. The hardest things to separate are an antibody and a misfolded antibody, or bispecific antibodies that are misaligned. And two molecules that look very similar that differ only in a few percent of their sequence. These are very difficult separations, but chromatography is very adept at these. But most chromatography relies on diffusion. So we've been using beads for 100 years, and these beads vary in size from a few microns per analytical scale to 100 microns or larger for other commercial production. So they're a very large process to use chromatography. High fructose corn syrup, which is used for soda pop, is widely it's purified. It's enhanced. That is, you can reduce and separate the fructose from the other sugars in an ion exchange column. And water purification. Everybody's home water softener, the one in your apartment building or the swimming pool or wherever it is, it relies on ion exchange. It's a chromatography process. And the difference is that at low concentrations, magnesium and calcium stick, and at high concentrations, sodium sticks. And you can manipulate the conditions and use it to separate different materials, even ions like sodium and magnesium, which are very closely related, but easy to separate. It's widely used. It's used at very large scale. Antibiotics at the 10,000 liter scale. I mean, these are huge production processes. So they all rely on beads. And a bead is a little sphere about 100 microns in diameter and is made out of plastic or silicon or could be cellulose even. And it has pores. And these pores are small holes that have dead ends. So the molecules are in the liquid phase. They come in contact with the sphere. They migrate into this pore by diffusion. It's a slow process. They may stick to the inside, and then they may come out later. We change the salt concentration or pH, for example, and then they come out again by diffusion. It's a slow process. So the speed of the process is defined by how big these pores are, how big the beads are, how fast we can flow, and the porosity of these columns is about a third of the column. If you can imagine, so we have these machines in Singapore that make fresh orange juice. And I watched the guy stack oranges in one the other day. I thought, well, that's just like a chromatography column. You have oranges, spheres next to each other, and you have space between them. And the space is about a third of the total volume of a chromatography column. And the diffusion, well, so you have convection flow between the beads and diffusion within the bead. But the problem is the large molecules and things like viruses and so on, they cannot get into the pores. They're just too big. And so the advantage of the beads is they have a large surface area and a small volume, just like filters, just like the filter in your coffee machine or your oil filter in your car. You want a large surface area in a filter in a small volume. For chromatography, we want a large volume surface area in a small volume. And we do that with these beads. But the issue is that they're not very good for some of these big particles like viruses. Viruses are used extensively for vaccines, for gene therapies, for cell therapies, also for exosomes, mRNA, DNA plasmids, all sorts of things are too big to fit in these pores. So the diffusion is not necessary, and so there's no point in using a bead. So the advantage of our system, and there are other competitors on the market have similar systems, goal is to have a large surface area in a small volume. And we do that by casting a polymer that has channels that allows the fluid to flow through these channels. And it's a small channel. It's about a couple of microns in diameter. And they are connected throughout it's the porous. It's like a sponge. So some companies are using membrane for separations, and they work fine. They have a low binding capacity because it's just a flat surface. So we tried to make it in 3 dimensions. It's like a membrane in 3 dimensions, and it has a large surface area. And then we can derivatize the surface area, put an ion exchanger on it, hydrophobic interaction, moiety, or some other thing, an affinity chromatography ligand. And that gives us the advantage of convective flow without diffusion and allows us to get a large surface area in a small volume, which leads to an enhanced purification.
What were some challenges you have encountered while developing this new technology?
And one of the challenges we call these technologies monoliths because they are a single structure. So the difficulty with the single structure, in the early days of beads chromatography, one of the main materials still used for making beads is agarose, which is a type of sugar derived from seaweed. And one of the challenges was that under different salt conditions or pH conditions, they shrink and swell. And some of these media were very soft, and they could swell to the point where there was no flow through them. And so the answer to that was to cross-link the agarose to make it rigid. So they did that, and that works fine for beads, but it doesn't work so well on a monolith because, again, we're looking at porosity, and porosity is the key factor. For convection in a packed column of beads, you're a maximum of about 36%. But for convection in a monolith, we can go up to 90%. That's wonderful. That means we can flow at very high flow rates and get a lot of surface area in a small volume. If it's not cross-linked so much, then it has a tendency to shrink and swell. And people don't ever talk about this, but we have found this to be a significant challenge. But we're very fortunate in Singapore. We have a lot of smart people in the country, and we have smart people working on the problem also in Europe, where our advisors are located. And we've solved the problem. We've come up with a way to handle the shrinking and swelling without it having a negative impact on our performance.
This is exciting. It's definitely an exciting technology, which will be a game changer, in particular for new modalities. Now we know that the biotech industry wants to be innovative, but actually, if you dig deeper, usually it's not that innovative. Other industries are much faster when it comes to adaptation to new technologies. So I would love to hear from you, Scott.
There's a reason for that.
There's a reason for that, yes.
And the reason is that drugs, unlike every other product, except the only one product I've found that has the same issue is nuclear reactors. Every product must have a license independent of every other product. If you make TVs, you don't need a license for every TV. You have a license for making TVs, and you can make 100 different models under that license. The same with cars. Yes, we do safety testing on cars, but that doesn't require a license. The government does not require you to submit data testing that proves it's safe, that the dose is right, and it's efficacious in randomized trials. What other products do that? Medical devices, yes, but only in healthcare. So that makes us very conservative, and it makes changes expensive. One of our key features is just like everybody else in this supplier industry, we want to be connected to the companies that are doing innovative work early on. So we provide samples of our materials to users early in their development so that they can test it and see whether or not it works for them. Our products don't work for everybody. Nobody's do, but they work very well in certain conditions. And we want to understand those conditions. We want to understand where it's best suited and then get into those companies early on in their development so that they don't have to make changes later, so that they have what they need from the beginning.
That's part one with Scott Wheelwright, diving deep into BioChromatographix International's revolutionary approach to chromatography and the science behind the monolithic media technology. In part two, we'll explore practical implementation strategies, regional bioprocessing differences, and his actionable advice for evaluating next-generation solutions. Got downstream processing development challenges? Book your free consultation at BrühlmannConsulting.com. Let's solve them together. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.